Good morning, everyone. It's really a pleasure to be completely out of my element and be here. So I really love this. I'm also going to add to sort of the discussions on sustainability in the broadest sense, but looking at sort of a different strategy some of us are pursuing. Very early days, but worth hopefully at least giving you sort of an update on where we are, where we're going, and why we think it's important. And I must say, I've learned so much from the first three speakers. Thank you all very much, because you've all approached the challenge ahead that we all face in really important ways. And we need everyone doing what they're doing and implementing what they're talking about. It's going to take the entire group here and beyond. So I'm uplifted already. So thank you for that, you guys. So hopefully I can leave you with nothing else today but a sense of what this field we call cellular agriculture is about, and where we think it fits as an alternative strategy going forward of feeding some of the foods of the future to this growing population. I'm going to start off with some very generic slides to make sure the language and the definitions are commonplace, and then get into just a few snippets from the research, because that's my home, right? Just to tell you what we're doing, why we're doing it, and more importantly, what we think the long-term impact can be. To set the stage, for those of you not familiar as much perhaps with cellular agriculture, which is essentially growing the animal, but without the animal, right? The same cells, but yet not on the farm. So I call it farming, PH instead of F for farming, right? But this field is really only 10 years old. Those of you who have followed this field, 10 years ago, Mark Post from the Netherlands went to the UK and on TV cooked the first cell-grown hamburger, if you remember this, right? It was ludicrous because it cost about $300,000 to grow the cells. These were bovine cells, to make a single hamburger to make this case. But at the same time, it spurred a lot of interest in the field. That is this possible? And my roots are in what is called tissue engineering, which is growing tissues for human clinical benefit. And so the segue to growing food is very simple, right? It's still growing muscle and still growing fat and all the things you need in meats and foods, but the metrics are completely different. One is cost, right? For human use, cost matters almost not at all if you're going to fix somebody's tissues or organs. When you're growing food, cost matters a huge amount. So all of us in this field that are sort of now trying to contribute to this have to rethink every step of our process, from the design goals to the components and how we go about bringing those together to grow protein-rich foods, meats, and so on. So I'm going to tell you some traditional ways we're doing this, but I'm also going to tell you some out-of-the-box ways, because I think we need really creative ideas going forward for the things like scaling, texture, and so on that we all want to get to. So that's a lot too much. I will add, though, just think about in 10 years, we've gone from 0 companies on the planet in this field I'm talking about to close to 200 companies today. Every single food manufacturer today, large manufacturers, are investing in this space, either their own activities in their own companies or they're supporting the startups in this space. You can go to any country in the world now and find companies starting up to build this infrastructure, particularly those with food security issues like Singapore and other areas. So they're really dedicated to looking at this possible alternative to really help what they're doing. And those of you who follow this as well, just recently, the last few months, you've seen the first 2 US companies approved by the FDA and hopefully shortly by the USDA to produce their foods and have them available to the public. And that happened a few years ago in Singapore as well already. So it's coming. The question is how fast and how much, and there's a lot of work to do, and that's the message I'll leave you with. So that's enough words. So you're all familiar with this, I'm sure, your favorite fast food. The point here is this is one element already on the shelves. Probably some of you have consumed them. I've consumed them. I actually like some of them. That has really taken a step forward. I use this not so much for these foods, because these are all plant processed materials, so they're not cellular agriculture, but a component to go into cellular agriculture. But I think what's stark to me is within 5 years, we went from plant-derived materials in sort of the cloak of vegetarianism to on every menu and in every fast food place in the world. And what that speaks to me at least, is consumers are looking for alternatives. What those mean, I don't know. I'm just saying they're hungry, pardon the pun, for finding other foods. And so we all want to do our job well to make sure we're providing what they need in a nutritious, cost-effective way. And so, again, those of you who know this space well, this is traditional plant processing, food processing, nothing new here. It's just done with some new additives like hemoglobin and so on that made the taste much better, et cetera. But it's still, extrusion, adding in plant materials, and on we go to make foods that are out there today. So, like I said, these are just one of the components. We call these scaffoldings, the materials that are going to support the cells for this field. The other segue that adds to cell ag are what I call alt proteins. You've heard this probably a lot of different ways. Think of this just as brewing beer, right? Or producing pharmaceuticals in a bioreactor at the center of this cartoon. Same thing here. This already happens, right? Cheese is produced because the rennin and other things are produced in mass quantities by this process. And you see this in all kinds of foods today, and there are companies dedicated only to producing these proteins, so B2B technologies as well. So this is a key element of what we're talking about today as well. So the plant-derived, the alt proteins, all into this topic of what I call cellular production or cellular agriculture. So here the equation is very simple, or I shouldn't say very simple, but relatively simple, right? We take an animal, the things we've been talking about today already this morning, and we simply do a biopsy from that animal to harvest cells, usually stem cells. I'll show you an example in a minute. And we then spend most of our time in the middle section here, expanding and expanding and expanding and expanding and expanding those cells, right? You need trillions to, huge amounts of cells to grow food. And so we do that in bioreactors. We'll talk about sort of futuristic ideas there to give you a sense of, getting out of the box here. And then we take those plant-derived materials we just spoke about, and those are the sort of architectures onto which we place those cells. So when we're done, we can grow or produce foods that you know and love already. And the idea here would be to make, at least initially, those foods identical to what you know and love today, but they're made only from the cells of those animals, not the living animal on the hoof, if you will, at the same time. I'll come to this in the end, unless I keep talking too much, but if you saw the news release just last week from Vow Foods, I don't know if anybody saw this huge meatball made from the woolly mammoth. Anybody read that article? Okay. That's ridiculous, okay? It's ridiculous because this is a museum meatball. It's not for food for human consumption. But the point is really important there, that you can take any animal on the planet. It doesn't have to be a domesticated livestock species, and you can harvest cells from that animal or from a prehistoric animal, and you can grow food that would represent that animal, meat from that animal, right? So for example, my lovely elephant at the bottom of these slides, right? That's the mascot for my university, Tufts in Boston, right? You would never grow elephant food domesticated for human consumption. But with this technology, you could do these things. All you need is a biopsy. The rest doesn't need the animal. So the point here is we don't have to think about this field even competing directly with cows and chickens. The idea is you can create all new types of pallets and foods that don't exist today, can't be approached today, and really can make a difference long-term in filling that 10 billion people gap that we're talking about this morning. So lots of things happening in the field, all early days. So please keep that in mind. So if you've never seen this process in action, this is what it looks like. Just like tissue engineering for human use, but now for food. Animals, we go out to the School of Veterinary Medicine at Tufts. We take our biopsies, grow up, isolate stem cells from muscle and fat, usually expand those out. And this is what they look like when you're done. If you've done your job well, you see beautiful muscle fibers that grow in the laboratory under controlled conditions. And if it wasn't a transferred slide set, you'd see these moving muscle bundles as well. So that's the process. But what's reality and where we need to get to? This is reality, and this is what we want to get to, right? We're nowhere near a steak. We're near muscle bundles. We can do that today, but we can't do this. And I'll show you one example of how we're trying to get there. So a lot of work to do to get to textured meats and so on, but we're making good progress on these factors so far. Lots of reasons. You've heard all this morning already, whether it's population, sustainability, et cetera, food security, food equity, all these issues play into this field that we're talking about, cellular agriculture. I want to give you a different perspective on this, and it's not what you've heard yet today. And that is, we can all sit here and talk about the science, and we can talk about what we see from our sort of window of importance for our fields and our constituencies. But I can tell you that I have never had students coming into my lab that are more passionate about these topics in my 30, 40-year research career, long time. But it's a pleasure, right? They're coming in because they care about sustainability. So they care about animal welfare. They care about food security. So I think, where this is going to go and how fast it goes, I can't tell you that. I can tell you there's such an undercurrent of drive from the younger generation that I think we all have to pay attention to it and make sure we nurture these processes going forward. So this alternative plays a role in all of our diets in the future. Now, with all that said, there are a million problems looking ahead, okay? And I just put a few of these on this slide. You probably can't read this. I'll just tell you there's issues of the cost, right? There's issues of scaling, issues of making those dense foods like meats, food safety, how are we going to do it better? Food taste and quality, GMO, non-GMO, et cetera. The list is long, okay? So I just picked out a couple of examples just to show you what we do today, to give you a sense of some of the things we're doing. But there, we could spend the entire half hour I have on one of these topics. That's the challenge today. So just so you're aware, there's lots to do, which also is great for the research lab. It makes it a very exciting time. But let me start off with something that's really important that cellular agriculture can provide that you can't do simply other ways, and that is you have direct access to cells, right? So you don't have to go through the ruminant. You don't have to go through other feeding strategies, metabolism, et cetera. We just go direct to the cells, right? Because we're growing cells in a bioreactor, so we can tell the cells what we want them to take up and therefore what their content is. And what does that buy you? That buys you control of composition, direct control. It buys you topics like personalized nutrition, and it buys you, in principle, healthier food. So I think this is one important aspect of this alternative strategy for foods of the future to think about. The other thing we do a lot of, and one example is to show you how we can directly feed. Here's where we grow fat cells. So I showed you muscle cells. So these are pig fat. So these are porcine isolated stem cells, adipose-derived stem cells. If you look at this plastic dish, it's just covered with fat cells. That's a 2D layer of fat cells that we grow in the lab. And then we scrape them up into a little ball. You can see that little white mass there. And we bind them together with traditional food binders, right? Whether it's an enzyme or an alginate or something. So you can go from very easy 2D to 3D fat bundles to use in plant-derived foods or cellular agriculture processes that we're talking about. But the important point here is we can control then the fat droplet sizes in those cells, just like you would see when you look at a pig fat in porcine meat. So we can grow the smaller or larger. That affects texture, affects taste, affects flavor, affects health. And then we can feed different types of fatty acids like omega-3s, and you can alter the composition of what those cells take up. So immediately you have control of the process in a way that's very difficult to do using the live animal. So I think this offers a future in what I call personalized nutrition through cellular agriculture as well. The biggest bane of existence in this field is the cost for growth factors, though. This is if you look at all the life cycle analyses to date, the cost for growth factors by far, everything else pales compared to this. So we and others are very actively looking at how to deal with that. And the two areas we've looked at are serum, because obviously bovine serum is very expensive and a real issue to scale up food the way we're talking about here. And the other are the recombinant or protein-based growth factors to grow these foods, whether it's FGF, HGF, EGF, and all these others. So the question is, how do we solve that problem? So I'll show you a couple of quick data points to show you how far we've come in a very short period of time. And I also want to just mention, because I heard one of the other speakers talk about this, that the role of modeling and AI in this entire process is front and center and part of what we're doing as well with these growth factors. I won't get into the details today, but that's the future, right? Identifying the cell you're growing, identifying the receptors on that cell that need to be teased to grow, to replicate, and then differentiate, and then designing or picking out the right sequence of a peptide and a source for that peptide that's low cost, so you know immediately where to go with the right material. And so stay tuned for all that. That's all coming in the next few years. So here's something simple we did, right? We took our bovine cells. These are the bovine satellite cells that'll grow into muscle that I showed you already. And we take serum and we replace it with albumin. If you know anything about serum, albumin is the dominant protein already in serum, and it's a carrier. It really brings stuff in contact with the cells. And so are we all the way there? No, the black line is the cells growing on serum. So that's sort of the gold standard. And you can see these other two lines here are replacing serum with albumin. We're already doing really, really well as we grow and culture those cells time and again. And if we don't use that albumin, you see the cells lose their vitality over time, right? This is days in culture, my apologies, with cell doublings on the y-axis. So it works. It's not perfect, but it's a great step in that direction. And importantly, you see that we retain the stemness of the cells. That's what that graph shows you without all the details. And on the right-hand side, you can see myotubes, meaning the cells still replicate, are still stem cell-like, and when we're ready to convert them to muscle, they still form muscle tissue. So we've done a good job already of getting rid of serum. And the second one is to replace that albumin because it's still very expensive. It's a recombinant protein usually. So we use different plant extracts. And now we're doing even better. Now we're doing almost the same as we could have without serum and without albumin. And we still have all those features of the cells that I talked about. You know, and I won't have time. I think I'm already too far. So I'll just say that cost-wise, by using plant extracts, and you can see 4 of them that we spend a lot of time on here, whether it's soy, rapeseed, cottonseed, et cetera, this is really a good source for some of the raw materials to bring those costs down. Competition with food source aside, these are good places to start. There's lots of others you can data mine. So if I just summarize those last half a dozen slides, you see this progression. Just follow the black line, right? This is sort of where we started with stem cells in the lab 3 years ago, right? We were up at almost $200 per liter to grow cells. You're never going to grow food like that, right? In just 2 to 3 years, we're down, literally an order of magnitude reduced just with some simple substitutions. And again, many labs throughout the country and really around the world are working on this as well. So I see this curve continuing to decrease over time, where this media cost problem is going to go away. And I see the only real cost challenge ahead is scaling. And we'll talk about that in a minute as well. All right, I want to just make sure I intersperse some crazy ideas so you know that you have to think outside of the box, as I said. So here's another thought, right? And we heard a wonderful talk just before about there's a lot going on all around the world, not just in the US, right? And another piece of that is the fact that everywhere except westernized worlds, westernized nations, people eat insects every single day. Okay, may not be their prime source of protein, but it's certainly a big source of protein. We obviously don't do that in the US, and mostly in Europe, we don't. But we are very actively working on cultivating insect cells, just like the cow cells I showed you, just like the pig cells I showed you. And why are we doing that? Because insect cells are already known to be grown on media, we talked about media costs, where you don't need all those things that are costly, right? So I put the list here, no serum required, no specialized growth factors, little process control. So all the costs goes away. So as a nutrient-rich, really good source of protein, this is amazing. The problem is in the US, consumers will just throw up their arms at it, right? No one wants to eat insects the way we're talking about. Keep in mind, these will be cells, not whole insects. So people, unless you label it, right, that's up to the USDA and the FDA how it's going to be labeled. But if this becomes a thing, right, you would not know the difference, right? Because it would be muscle cells, fat cells, just the way we talked about cow cells and so on. So strong nutritional benefits already known for insects, There's insect farming booming in all around the world, as many of you know. That's the whole insect. We're not fans of that, but they're there. Flavor benefits are out there. These are well documented in the literature. So we see the initial goals here not for human consumption, right? Because it'll take a while for the consumer to get on board, at least we think. But for fish feed, for aquaculture, this is a perfect way, a segue to get away from feeding harvested fish to grow fish, right? Perhaps for livestock feed and perhaps for pet food. So these are the kind of initial markets we would see for this approach. And eventually, maybe for humans as well. We will be doing testing, so you're welcome to come visit. The other thing I want to mention is just the last thing on insects. I like to always show some data. So even if you just look at color coding here, right? Look at the left-hand column and it goes from green to red, and you look at this other column, it goes from green and stays green, right? This is an example where we took some insect cells, which is this column here, the second column, and we took some mammalian cells. We put them in a dish, separate dishes. We closed them up, we fed them, closed them up, and didn't touch them for a month, right? And we came back a month later, and you see all the insect cells are thriving. Green means they're alive and happy, and all the mammalian cells are dead. They're red and unhappy. That's an example, just a visual remembrance of how simple it is to grow these cells and how low cost it would be going forward. All right. The biggest challenge I see is scale up. I don't have time to go into all the issues here, but people talk about putting all the fermentation capability on the planet together on one place, and you'll still be probably 2 to 3 orders of magnitude below what you need to grow enough food to make any kind of difference in the food infrastructure out there, right? So this is not a trivial issue. And one of the banes of my existence, and maybe I'm the only vocal one about this, is too many companies are starting out emulating what you see in the pharmaceutical business, right? Where you have steel tank reactors, these are stirred tank, lots of plumbing, you can see all the plumbing, lots of process control. I don't believe, other than mesoscale, I don't think you'll get to mega scale with this. It's just too expensive, too much requirements in terms of energy and so on, even just to make these structures. The good thing is there's companies now started in cell ag space dedicated to building the next generation of bioreactors that is completely divorced from this style, because they understand the challenges ahead with economics. So hopefully those will come to the fore. I will tell you one of the strategies we have, again, out of the box, so stay with me on this, is a knitting strategy. And I know it sounds trivial, but the idea is literally to knit your 3-dimensional, dense, protein-rich meats of the future. And how do we do that? We were very fortunate. Merck gave us a large grant over 3 years to demonstrate proof of concept, and we're about halfway through. And the idea is very simple. We simply need edible fibers as the core of what we're doing. We already have edible fibers. They exist. They're out there. They're available in large quantities. And we grow our muscle cells along those fibers, or grow our fat cells along those fibers. So this is just 1D tissue engineering, right? Fiber-based tissue engineering. No limitations to fusion, anything now. It's very simple to do. And when you're done, you have muscle-laden fibers, and then those go into traditional textile engineering as a downstream process. Twisting, weaving, knitting, layering, whatever you want to do. So the infrastructure here is just really this first bioreactor, which can be arrayed into millions of these strands, continuous process, and so on. So what the costs are really going to be, I can't tell you. The process does work. I can tell you that because it works in our lab. The question is, is it going to be scalable into manufacturing technology ahead? Might be a few years, might be a decade, I don't know. But at least it's another way of thinking apart from stirred tank reactors that might get us to a scalable technology that could have some impact on future foods. And I would add that this solves the densification problem. That's an inherent limitation, because you can twist and pack these tightly. Even if the cells die, it doesn't matter at that point. And so this, again, overcomes some of those texture issues you'd like to see with these technologies as well. All right, very briefly, 5 minutes. I just picked this out for the fish world, right? For fish world, because we've been very active in the fish technologies as well. If you know the challenges with fishing and fish-related meats and protein-rich foods, you can go down the list, and you can do this for any species you want, right? But depleted fish stocks, you already know. Harvesting fish to feed fish, kind of a bit crazy, right? Micro and nanoplastics have become really an issue lately, rising costs for fish, healthy fish, and then the accumulation of toxins and chemicals in the fish ecosystem. So there are lots of problems. When you buy fresh fish, do you really know how good it is for you anymore? So the idea is cellular agriculture can address all of this with controlled environments, tunable nutrition we talked about, and really scaling the ways we're starting to talk about as well. So we see lots of opportunities. And one example of what we do in the lab, just so you know, we have lots of fun too, right? It's important in the research. So we go on field trips out Gloucester, Mass into deep sea fishing. And fortuitously, a couple of years ago, we caught a mackerel. We obviously grew the cells, we harvested, did biopsies, expanded those cells, made sure they were mackerel when we were all done, so did our sequencing, right? And then we were able to what we call spontaneously immortalize those cells. So non-GMO, just culturing these cells time and again, and eventually you get past 50 doublings and they just go and go and go, which is really nice to see. And that allowed us to spin off this technology into a startup company, which is now trying to grow three-dimensional mackerel and other fish tissues for eventual human consumption. In other cases, we're not so lucky. In other cases, unlike the mackerel, where it's spontaneously the cells immortalize, sometimes we have to use GMO basis, right? So we use genetic tools. So here's an example where we took our bovine muscle cells I told you about earlier. Here we use specific genetic modifications to make these cells immortalize. You're going to need immortalized cells for the scaling we're talking about. And you can see how well it works. Here's the black line are the non-immortalized muscle cells I told you about earlier. They last for about 50 doublings. And then they start to 50 days, and then they start to peter out, right? They're no longer active. They go through senescence like any of our cells go through. Whereas if we immortalize them, they just go on in principle forever, right? So now you have a cell line, and in terms of food quality, consistency, quality control, et cetera, these cells could be banked, shipped around the world. Anybody could use them to grow similar kinds of food. And so we've deposited these in our muscle cell and our mackerel cells in databases so people can use them for their research and for making foods and so on. Importantly here, you notice the doubling times, even with the immortalized cells, don't go down. They stay pretty good. So we're in good shape here, about 15 hours for these cells to double, and they still make muscle, et cetera. All right, last bit in my last few minutes, really quick. I think the whole other area, we talked about direct access to cells. The other area is really if GMO is going to be something consumers accept. We don't know that answer. I would think there'll be a segment that will and a segment that won't. So we do the research for both. But with GMO, it opens up incredible windows of opportunity for this alternative food source of the future, whether it's the cost issue, increasing value through other things like shelf stability, which I'll talk to you about. We just heard about wasted food a minute ago, as well as nutrition. So one example we've published, and we have others coming, is very simple and shown here. This is using synthetic biology tools to take those muscle cells, bovine muscle cells, and make them into carrot-producing cells, if you will, right? So we import a pathway, a 3-gene pathway, that allows what's in the muscle cells already to now make vitamin A. So now you have muscle cells growing that look a little bit orangish, not too bad, right? But they're full of antioxidants now. And why does that matter? That means they're healthier for us to consume in principle, right? And these survive cooking as well. That's all published. And more importantly, we think this is a way to even have existing meats like this that could be more stable on the shelf, meaning less waste, less food waste at the same time. So there's a zillion examples here where we could engineer the cells to be proactive in different ways. And so this is the future, right? Not only what we've talked about, but can you take foods from the past? You know, we just saw the woolly mammoth. There's going to be other things you'll see as well. I talk a lot about therapeutic foods, segments of the population that need very specialized foods, where you could engineer those foods and have those delivered to homebound folks or specific populations with specific diseases where those foods are really highly tailored based on cellular agriculture principles. We talked about personalized foods already as well. And I think food can be turned into, with what we're talking about today, kind of the not just growing putting bread machines in a kitchen, right? But you can literally have 3D printers and other fun things for kids in the family to do to not only cook the food and prepare food, but actually make the food at the same time. So it becomes part of the culture of the family and the community at the same time. So these are things, I like to speculate, but that's what the future may hold with this. Briefly, just to conclude, one, I want to just acknowledge the Department of Agriculture. NIFA has been amazing supporting what we're doing. It's a consortium of universities led by ourselves, along with Virginia Tech, UC Davis, MIT, Virginia State, and UMass Boston. But we're working on educational programs. So if anybody has interest or students with interest, just have them contact me and we'll get them the right information. We collaborate with lots of folks out there and we try and build technology for others to use. I want to also mention we have an industrial consortium that's very active. We have 9 founding members, traditional food companies to startups in the space. You can see all the names here. And the companies decide what the needs are in cell ag, and then faculty at Tufts deal with how to develop projects and research to help those companies towards those goals. So that's something happy to talk more about if you have interest. I want to just thank amazing collaborators all over other funding sources and my group as well. Thank you very much.